Okay, let's dive right into this explainer. Today, we're exploring how a tiny, isolated group of individuals can literally trigger massive evolutionary divergence. We're looking at a completely fascinating biological phenomenon where the sheer power of isolation just rewrites the genetic destiny of an entire population. It's a wild story of survival, pure random chance, and the total breaking of mathematical rules.
So, what exactly is the founder effect? Well, it's a very specific, kind of dramatic form of genetic drift. It happens when a very small group of individuals completely separates from a much larger parent population to go establish a brand new colony. The absolutely crucial takeaway here is that because this new group is founded by merely a few organisms, it inherits and carries only a microscopic fraction of the original population's massive genetic variation. Here's our roadmap for today. We'll start with the breakaway population, move into random sampling error, hit the isolation effect, explore breaking evolutionary rules, and wrap up with some real-world examples. Section 1. The breakaway population, the inciting incident. There is a stark, almost terrifying contrast between the massive diversity left behind and the incredibly limited gene pool of the new founders. See, in a huge mainland population, rare traits stay uncommon simply because they're heavily diluted among thousands or millions of individuals. But when a handful of individuals migrate away, they represent an exceptionally tiny sample. That means an immense amount of genetic diversity is just permanently lost. The descendants are going to inherit absolutely nothing beyond what that tiny group brought with them. Picture this timeline. 0 hours, a powerful storm hits the main flock. Within a single day, just a few surviving birds are blown hundreds of miles off course to a remote island. By week 1, absolute geographic separation from the ancestral population is permanently established. This naturally occurring example shows exactly how violently and quickly a founder event can lock a few survivors into a brand new genetic trajectory. And once they're isolated, their future is heavily dictated by random sampling error. It's basically a genetic lottery. Section 2. Random sampling error. The genetic lottery. Think about how a rare trait acts in a massive parent population. Let's say we have this deeply rare blue allele that exists in just 1% of the mainland group. Because there are millions of individuals, that 1% stays firmly at 1%. It is quite literally a drop in the ocean. But over in our new founder colony, strictly by random chance, that incredibly rare blue allele suddenly surges. If just one of our 4 storm-blown birds happens to carry it, boom! Suddenly it represents 25% of the entire island's gene pool. The gene itself didn't change at all, just the mathematical reality of the population size. Biologists call this mathematical quirk allele amplification, and it is the core biological mechanism driving the founder effect. Because of that exceptionally tiny starting sample, a trait that was extremely rare can dramatically and rapidly surge in frequency. Over the next generations, absolute isolation and mating within this tiny group will just amplify those once rare traits even further. Section 3. The isolation effect, endogamy and divergence. So, how does this actually play out over time? It's a pretty ruthless step-by-step process. Step 1. Complete isolation. The evolutionary force known as gene flow from the mainland just completely halts. Step 2. Restricted meeting. Because the absolute size of the population is remarkably small, meeting inevitably happens among close relatives. Which leads to step 3, endogamy, or inbreeding. This vastly increases the mathematical probability that offspring inherit the exact same alleles directly from shared ancestors. And finally, step 4, a massive increase in homozygosity, which means the population's genomes become incredibly, almost dangerously uniform. Section 4. Breaking evolutionary rules. Dismantling Hardy-Weinberg. What's really wild is that this survival story systematically dismantles the Hardy-Weinberg equilibrium. In biology, Hardy-Weinberg is the baseline mathematical rule for a population that is not evolving. But our founders absolutely wreck the 3 main assumptions. First, there's no infinite population size. Founder populations are inherently tiny, meaning fewer chance dictates allele shifts. Second, there's no random mating. Desperately limited partners encourage continuous, non-random inbreeding. And third, absolutely no migration. Extreme isolation shuts down outside gene flow entirely. And here is the ultimate takeaway. Because these core assumptions are utterly broken, the allele frequencies continuously mutate and change over time, forcibly producing true evolutionary change. That is exactly how a tiny group of castaways rapidly transforms into an entirely new biological reality. Section 5, Real-World Founder Examples, the Amish and beyond. To really grasp this, let's look at real-world populations. Take the North American Amish, for example. They represent a culturally and socially isolated group. Historically, an exceptionally small number of their founding members happened to carry deeply rare alleles for specific genetic disorders. Because the community stayed so remarkably isolated and strictly practiced endogamy, the frequency of those disorders became vastly higher there than in the general public. On the flip side, looking at geographic isolation, like our remote island birds, just a few colonizing organisms can easily establish a brand new population totally distinct from its mainland ancestors.
Ultimately, though, these founder populations face some incredibly severe long-term consequences. We're talking a drastic, permanent reduction in genetic variation. By pure chance, fatal, harmful recessive traits can become overwhelmingly common. This inescapable inbreeding actively exposes deadly genes, leading to inbreeding depression, which basically means drastically reduced survival rates and fertility. Worst of all, it critically reduces their ability to adapt to environmental changes. But serving as a major mechanism of genetic drift, it also plays a vital role in